We're going to go over blood typing today, and we're going to start by first figuring out what an antibody is and what an antigen is. So, first of all, let's define what an antibody is. Now, these are sometimes also called immunoglobulins because they are proteins, globulins, that the immune system makes. And typically the cell that we think about with antibodies is the B cell. And the B cell, when it is activated by some sort of pathogen, it will activate and turn into a plasma cell. And these plasma cells are able to then make these Y-shaped antibodies. And these antibodies are proteins that then can be released by the plasma cell to then go and attack, for example, a bacterium. So let's say if this is a bacterium right here, the antibodies will bind to the bacterium and help to neutralize it and help to also activate other white blood cells, such as macrophages, to come in and to eat the bacterium. So the antibodies attached to the bacterium help this macrophage do its job, which is going to be phagocytosis, otherwise known as eating of particulate matter, in this case the bacterium. So the question is, and we're going to talk about this in more detail when we get to the immune system, but the question is, how does a plasma cell get activated to make antibodies? So one thing that happens is that bacteria have little bumps on the surface of their membrane. And these little bumps, made of proteins and sugars, are known as antigens. So there is an antigen right there on that bacterium. And basically, these antigens are foreign to your body, and your body recognizes this and realizes that it can make proteins, known as antibodies, that are sticky and are specifically sticky to those antigens, right? And so basically, whatever antibodies this B cell makes are going to be specific for the antigens that are found on that bacterium. Now, another trick to remembering this is to actually know what this word stands for, because this is something that is often not talked about. So antigen is actually a short term word for a longer word known as an antibody generator. And that makes complete sense based on this conversation, because now, if we look at this, we realize that these are in fact antibody generators, right? They generate the antibodies. Their presence inside your body will end up getting your immune system to make these immunoglobulins / antibodies that are specific to that antigen. So what they've done is they've taken this part of the word, anti, and this part of the second word of generator, right? And they've linked them together in order to make this term antigen. So hopefully that will help you remember to not get these two terms confused, because that does sometimes happen where students are not sure which one is the antigen and which one is the antibody. So the second thing to understand before we start our blood typing is to also understand that there are different classes of immunoglobulins. And there are several different classes, and we're just going to refer to two of them for this particular lecture. And what we call them is I, G, and then G. So we've got IgGs as one type, and that's the type that is that Y shape that we just drew, which is the most famous depiction of antibodies, is just that Y shape, and that's the one you're typically going to see in most pictures. But there's another one that we have to talk about for this blood typing, known as IgMs. And IgMs, the only difference is that they're bigger. And the reason why they're bigger is because you take 5 of these and you link them together at a center point. So there's 1, here's the center point. There's 2, 3, 4, and 5, making this pentameric antibody, which is obviously going to be 5 times larger than this IgG. Now, the reason why this is important is because when we talk about blood typing, we do have antibodies that we have to talk about, and the A and the B antibodies end up looking like this one. This is critical to understand that this antibody right here, because it is so large, cannot cross the placenta, which is, the placenta is of course the tissue that ends up providing oxygen and nutrients, right, from the mother to the child during pregnancy. And what is important about this placenta is that if you imagine that there is a mother's blood vessel network on one side, and then there is the child's blood vessel network on the other side, right? And right, we want to think about oxygen moving from the mother's side to the child's side, right? The two bloods do not mix, right? So there's no mixing of blood here. And so we should not have certain things crossing over. So whereas oxygen can cross over, it can just diffuse through, this IgM is too large and should not cross over and will not cross over into the child's bloodstream. But there is another type of antibody that we do find in blood typing that we're going to talk about, which is the Rh. And the Rh is a special one, and it is one that can be acquired, and we're going to see how this works exactly shortly. But basically, if the mother has this Rh, then the Rh is small enough, because it's just that single antibody, Y shape. It is small enough to where it can cross over into the child's bloodstream. And if it does, it can start attacking the child's RBCs and causing a type of anemia called hemolytic anemia of newborn. So we're going to talk about how we want to avoid this from happening. To understand blood typing, what we want to do is draw out 8 red blood cells and we want to start labeling them and also understanding why we call them as such. So let's make a key up here, and for this lecture, I'm going to make the A antigen a triangle. I'm going to make the B antigen a circle, and I'm going to make the Rh antigen a square. And that's just what I'm doing. Obviously, these shapes don't actually exist. We're just using these shapes as a mechanism to understand what we're doing. So please understand that these antigens here, whether it's the A or the B in this case. So first, just focus on the A and the B, and don't worry about this Rh one right now. So when you think about these two, think about the fact that a human that is born will possibly have an antigen A that sticks out of its red blood cell. These are just proteins that stick outside of the phospholipid bilayer of these cells. Now, if you recall from your phospholipid bilayer studies, there are proteins embedded within the phospholipid bilayer of cells. And specifically for blood, what we have are these special proteins that stick out. And so the individual is basically born with these. And the reason why they're born with these is because their DNA has the genes for these, and therefore that DNA is expressed through transcription/translation onto the surface of these cells. So let's just say that this is an individual, right? And this individual has this blood. And let's just say that that individual has the DNA gene for the A antigen/protein. And therefore this individual ends up putting that protein onto its cell membrane. And this is basically just a name tag for that cell. If that's the case, what we say is this person is type A. They have a type A blood because they are depicting this antigen on the surface of their cell. If you recall, antigens are originally thought to be on bacteria, so it's strange to call this an antigen because it is a human cell, but we're going to understand very shortly why we call these antigens. So this is a type A person. Now, over here, let's draw the B antigen. And if we draw the B antigen, then this person would have type B blood. It is also possible to have the DNA and the genes for both of these. And if that is the case, then this person would have both the A and the B on their membrane of their red blood cells. And at this point, we would of course call them AB. It is also possible to not have either the A or the B, which means that there is nothing regarding A and B on the surface of this cell. And so in this case, we call it type O blood. So this is the way to start the conversation. And then what we're going to do is we're going to add on to this as we go along. So let's box these in. And what we're going to do is we are going to also draw what is going on inside the plasma of these bloods. So plasma is, of course, the liquid of blood. And the liquid of blood, as you have learned, is composed of not only water, but also of electrolytes. It is composed of proteins. It also has sugars. It has gases that are dissolved inside of it. And for this lecture, we're just going to talk about what immunoglobulins are going to be found inside the plasma of each of these. Right? So the plasma is, of course, going to be found outside of the cell, right? It is the liquid that is outside. So the way that this goes is that if this person has a type A blood, then the antibody that is going to be inside the plasma of this person is going to be the B antibody, and it is an IgM. So it has that pentamer shape to it. Now notice the way I drew this. I drew this as a circular, right? Instead of that Y shape that I started with, I ended up making the sticky ends of this immunoglobulin circular in shape, because what I want to show is that this would actually be sticky to this circle right here. And it would not be sticky to that triangle right there. So what we want to do is make the shape of the antibodies match the shapes of the antigens. Right? So this is an anti-B antibody. And notice why this is so important. This is a person's blood. Antibodies attack antigens. And just realize that this anti-B antibody cannot attack that triangular shaped antigen, which means that this person's plasma will never attack its own red blood cells. And that's exactly what we want. If we had a situation where you had an anti-A antibody inside the plasma of this A blood person, it would attack and this person would not survive. So for this person to survive, there cannot be a match of the antibody to the antigen. I hope that makes sense. So if we do the same thing for B, what we do is we draw an antibody that instead has that triangle sticky end. And of course, this triangle sticky end would match the A antigen, but for this type B blood person, this is good because this anti-A antibody will never attack this circularly shaped antigen, which means that this person's plasma will never attack this person's red blood cells, which is what we want. So now think about what we would have to draw here in this picture for this plasma of AB blood. I'll give you a second to think about it. Basically, you don't want the plasma attacking the antigens. So if you thought about this enough, you would have realized that we cannot have either of these inside the plasma of an AB blood person. On the other hand, for a type O blood person, think about what you would have. I think you know what it would be. You would end up having both. You would have the anti-A with the triangular sticky ends, and you would have also the anti-B with the circular sticky ends. And so this is the way to start understanding how blood typing actually works. So once you've understood this basic idea, the next thing is to now introduce the Rh antigen. And this is a little bit different, and it's a little bit tricky. So you will have to think about this differently. And the reason why is because if you take this individual that's born with the A antigen sticking out of its RBC, this individual is also born with this ability to produce the anti-B antibody. So this is how this person is born. Same thing with this person, and same thing with this person, and same thing with this person. The plasma will match. Okay, at birth, the plasma will match in this way, the way that the RBC is being depicted. Now the Rh is a little different. So down here what we're going to do is we're going to draw the same thing that we drew already. We're going to have the A, the B, the AB, and the O. And we're also going to draw once again the plasma antibodies. And make sure you do this with me. Don't just watch me do it, because by drawing this out, that's how you're going to learn, and that's how this information will stick better. So right now we've just basically redrawn what is above, but now we're going to alter it a little bit. So now what we're going to do is we're going to put in this Rh antigen, which is this square shape that's going to stick out on the surface of these RBCs. So there is one right there, and I'm going to do this for all of them. And the important thing here, and this is what I was saying before, that with the Rh you have to think about it a little bit differently. And this is how. So in this case, notice if I have the Rh antigen, all I do is I change this slightly and I put a positive. So this is A positive, and that's what this blood type is, which makes this one A negative. This one is B with the Rh antigen, it will be B positive, which makes this one up here B negative. Same thing here, AB positive, AB negative. Same thing here, O positive and O negative. And now what you have to do is you have to also talk about the Rh antibody. And here is the big difference. The big difference is that obviously the positives cannot have Rh antibodies inside the bloodstream because they will attack themselves, right? So then you might think to draw in the Rh antibodies into the plasma of the negatives. And we will. And so we will in order to make our lives a little simpler. However, just realize that these individuals are not born with the Rh antibodies. That is the big difference. So we'll put a little star here. Not born with Rh antibodies. Okay, not born with Rh antibodies. So then that means that these Rh antibodies have to be acquired. How could we acquire an Rh antibody in this person's plasma, you might ask? This person would have to be exposed to a positive blood type. So just imagine that this person here that has a negative blood type is then exposed to this person. In other words, the blood mixes, and this A positive RBC ends up entering into this person's bloodstream. At that point, the immune system of the negative person will manufacture antibodies using those B cells that turn into plasma cells, and will in fact manufacture them and will end up building them, and those will then be found inside the plasma of that person. Now notice how I'm drawing them. I'm not drawing them as IgMs, I'm drawing them as IgGs. And that's a big other difference here. These are the ones that are implicated in the hemolytic anemia of newborn because these are small enough to cross the placenta, as we're going to see shortly. So for right now, just imagine that in every single case, you have some mixing of blood and some positive RBC enters a negative bloodstream, and the negative individual's immune system manufactures this antibody. And you do that for all of them. And so this is a way to simplify a little bit what we're going to do here. We are going to assume for any of the questions that we have regarding if there is compatibility between blood types, we're going to assume that this Rh antibody is in fact present in every single negative individual's blood plasma. However, it is important for you to know, for your knowledge, that this is not something that will always be there unless there is some mixing of blood, which can happen from, for example, sharing needles, from sexual intercourse, from childbirth, anything that can end up getting bloods to mix. So you have to have this next to you, therefore, when we now talk about compatibility of bloods. And so I'm going to move this to the side, but you should have your own next to you for the next conversation that we have. So now what we're going to do is we're going to ask ourselves, in each instance, we're going to give examples, and we're going to say that we have a donor. In other words, somebody who donates their blood, and we have a receiver or a recipient. Somebody who receives the blood. And for the donor, you have to understand that for the donor, what we're going to do is we're going to centrifuge the blood, which was talked about at the beginning of chapter 19, where we spin the blood around and we separate it into its components. And if you recall, there is the plasma, there is the buffy coat, and there's the RBC layer, right? So in this vial of blood, you have those three layers, right? You've got the plasma, you've got the buffy coat, and then at the very bottom, you're going to have the RBCs. Right? And basically the plasma is the one that has the antibodies inside of it. The buffy coat is the one that has the white blood cells. And the RBCs are at the bottom here. And so what we do with the donor's blood is once we separate it, we just take these RBCs out and we donate them to the recipient. And so when you think about the donor's blood, all you think about is the RBC. That is a very important concept. When you think about the recipient's blood, you have to take into account the plasma, because it will be the plasma that does the attacking of the RBCs that are coming in. So with that in mind, let's go ahead and do an example, because that's how you're going to understand all of this. So the way to do this is just to do as many as possible. And so let's go ahead and say, what if we take, right, looking at our picture that we have, that we drew already, what if we take an A negative person's RBC and we donate it to an A positive's person blood, right? So we're going to take this RBC, only the RBC, because remember, we centrifuge and we just take the RBCs. And so you don't worry about the plasma when it comes to the donor, but you just take the RBC and you put it inside this person's blood. And you ask yourself, can the plasma attack? And so what I do for all of these is I actually draw out little pictures for myself. So here's the first one. The donor is going to be A negative, and the recipient will be A positive. And so I ask myself, what does this RBC look like? So I just draw it shorthand. I draw the RBC, and I draw the A antigen, and then I ask myself, can I put it inside this person's plasma? And then I draw out this person's plasma with the circular sticky ends. Right? Let me just show you once again, just to make sure that you're with me here. Right? I drew out this RBC. I did not worry about the plasma for the donor, just the RBC. But then I asked if I could put this inside this plasma, which meant that now I had to draw this out, right? I had to draw the antibody found inside the plasma of this A positive person. And that's why I ended up drawing this. So hopefully you're with me on that. And now I have to ask myself, can this sticky end of this antibody attack? Well, this sticky end is circular and this antigen is triangular. So will there be a match? Will this plasma attack that RBC? No. And so can I donate that negative blood to this positive person? Yes. Okay, let's do another one. Let's reverse it. Let's say that I have an A positive person as the donor. And I want to ask, can I donate this RBC to the plasma of an A negative person? Now I have to draw the antibodies of the A negative person. So let me look at that sheet again. So I'm asking, can I take just this RBC and introduce it inside the plasma of this person? And remember, we will assume that the RH is going to be there, right? Even though this person may not have it, right? Because they're not born with it, but they can acquire it if there is blood mixing. And we will assume that there has been, and so we will assume that this RH is present. So think about it. Can you take this cell and introduce it inside that plasma? Well, the way to figure it out and not to make a mistake is to go ahead and draw out the plasma. So I've got the RH antibody, and I've got that anti-B antibody, right? So this is circular. I'm trying my best to make it circular. And this is square. So then I ask, can either of these attack either of those? Well, the circular cannot attack the square, it cannot attack the triangle, so fair enough. But what about this one? Well, this one is square, right? So that square one can attack this square one, right? So if you, let's just bring it into the picture, right? You take this cell and you introduce it, right? This will rotate, it will turn around, and it will stick to that square, so there's a match. In other words, the plasma will attack the cell. And so can we do this? No. It is not compatible. We cannot donate a positive person's blood to a negative person's blood. So let's continue then. Let's do another example. Can I give an A negative blood to a B negative blood? And so here I draw the cell, but here I draw the antibodies. And so looking at this sheet again, I'm basically asking, can I take this RBC and put it inside this plasma? All right, so I have to draw that RBC, and then I have to draw this plasma here. So let's go ahead and draw that. I've got the square-shaped RH antibody, and then I've got the triangular shape. Oh, having a hard time drawing that one. Should be 5 of them. 1, 2, 3, 4, 5. Okay, good enough. And these are supposed to be triangular shaped sticky ends. Let me make sure I have that. Triangular shaped sticky ends. And my question is, if I insert this RBC inside this person's blood, can their plasma attack? And of course it can, because this is triangularly shaped, this A antigen, and this is triangularly shaped antibody, right? So this will attack that, so no, I cannot. Which means that you cannot give type A blood to a type B blood person, right? And of course, if you think about it, right, the same is going to be true if you swap these. So let's go ahead and swap them, but let's, in this case, do these two. Let's take B positive and try to donate it to A positive, and let's see what happens. So we're going to take this RBC, and we're going to try to insert it inside this plasma and see what happens. So B positive, can I give it to A positive? The plasma of an A positive person will have the anti-B circular sticky ends. And so now if you ask yourself, right, can this go to here, right, this sticky end will attack, right, that circular antigen. So no. Try to think about some patterns that are going on here. Once you practice these enough, you will find some patterns. So that, for example, if it's type A, negative to positive, it's a yes, right? And that would be the same if you replace the A's with the B's, right? Or if you had AB to AB, or if you had O to O, right? Notice that if you got a positive, right, going to a negative, that's a no, and that would be a no also for B to B, right? B positive to B negative, that would be a no. AB positive to AB negative would be a no. O positive to O negative would be a no. So those are some patterns that you can figure out once you do enough of these. And then, of course, when you mix the letters up, you also get no's. So now let's go ahead and look at what happens with some examples of ABs and Os. And so, for example, let's do AB negative, right, as the donor, and the recipient, let's say it is O negative. So again, right, we always go back to this, and we want to know if we can donate this RBC into this person's plasma. Now, at this point, you should be getting pretty good at this, to where you can just imagine dragging this into that and wondering what would happen. So let's go ahead and draw it out anyways. For your test, I do suggest you draw it out if possible. So here's the cell for the AB. I've got a triangle, I've got a circle, and then the O negative plasma is going to look like that. Like this, and also with the triangle sticky ends like that, right? So think about this. I mean, this right here, this O negative person has a lot of attackers inside its plasma, right? So in general, if you think about O negative, we're going to have a hard time finding a match for a donor to donate to O negative, right? And so just realize if we take this AB negative person, there's a triangle there, there's a circle there. This one will attack the circle, this one will attack the triangle. So now you've got a double attack. So definitely a no, right? And on the other hand, let's say that we have O positive to AB positive. Well, the O positive, if we're looking at this, right? We're asking if this RBC can be introduced into this plasma. Try to use your imagination. Can this plasma attack? Well, there's nothing there, right? So, right, I think you know what's going to happen here. The O positive RBC is just going to have the square sticking out, and the AB positive plasma will have no antibodies in it, right? And so nothing can attack that. And nothing can attack anything that comes in. So this is a yes, but a good rule to remember for AB positive people is that because they have no antibodies inside their plasma, they are the universal recipients, right? They can accept from anybody because they don't have anything to attack with for anything at all. If you take, let's take the other extreme here. Can you think of an RBC that is basically empty of any antigens? This one right here, right? So O negative blood, let's see. If we take O negative blood, right? Let's draw it out. Here's the RBC. It has nothing sticking out of it. And, right, it doesn't matter what we put in this plasma here, right? We can put triangular sticky ends. We can put circular sticky ends. We can put square sticky ends. Doesn't really matter, does it? This RBC can be introduced into any blood, and so the O negative person is known as the universal donor, right? So this is a yes. So I do suggest making your own, doing some of your own examples just for practice, just to make sure that you have understood this. This is the way that I do it and my method of doing it, which I find does not give me any room for error to where I don't make any mistakes. There are some general rules that you can find on Google that tell you just some general rules for this, but I would suggest that you don't skip to those rules without first understanding it, because understanding it is really much more interesting than just memorizing rules to get the answers right on tests. And so test questions will typically say, can I give AB negative blood to an O negative person? Yes or no? Or the question might ask, what kind of bloods can I donate to an A positive person, right? And it might have like a list of multiple choice. Or which one of the following can I not donate to an A positive person? And then it would be a multiple choice question, and you would have to pick the best answer. But sometimes questions will be phrased in a way to make sure you actually understand it. So a question might ask you something like this. The reason why I cannot donate A negative blood to a B negative person is multiple choice. Because of the Rh antibody, or because of the anti-A antibody, or because of the anti-B antibody, or all of the above, right? And so, of course, the answer would be because of the anti-A antibody. So it does get confusing. It does take some time to get good at this stuff. So take your time, draw these out, make some charts for yourself, and you should be okay. The last part of this lecture will be to understand hemolytic disease of newborn. And the way to understand this is to remember this very important fact right here, that we are not born with the Rh antibodies. And if you remember this, then you can use this understanding to understand hemolytic disease of newborn. The second thing to remember is that we do have these different immunoglobulins. And we've got the IgGs, which are small enough to cross the placenta, and that's the Rh. And then we've got the IgMs, which are not able to cross the placenta because they have these 5 branches, which make them 5 times as large as the IgGs. So when it comes to hemolytic disease of newborn, we do not worry about the A's and the B's, but we do worry about the Rh. So the way to talk about this then is to imagine a woman and a man having a child. Let's just say mother, father. Good enough, right? And what we do is we imagine, for example, that the mother is A negative. Okay? And we imagine the father, let's say, is A positive. And now what we do is we imagine that the child is also A positive, like the father. And this is child number 1. So first child. And so what we do is we realize that this child here, its RBC is going to look like this with the triangle and also with the square, right? And that the mother, let's just assume for this discussion that she has not made the Rh antibody yet. And so right now, currently, right, during this pregnancy, all she has is the anti-B antibody for now. Right, so that's her plasma. And this is the child's RBC. Now, during childbirth, what's going to happen is, so the placenta, we don't get mixing for now, everything's okay. But at childbirth, there may be some bleeding that takes place, right? Some of the mother's tissue may get ruptured during the childbirthing process. And in that process, you might get some RBCs from the child introduced into the mother's bloodstream. So this will not be happening during the pregnancy, right? The bloods are not mixing. The placenta, the blood is not mixing. However, during childbirth, right? Then with some bleeding, you may end up getting an RBC that does actually get inserted into the mother's bloodstream. So now at this point, her immune system will react to this antigen, which is foreign to her, right? Because her RBC, after all, her RBCs look like this. So they're a bit different, right? They're A negatives, right? Whereas this is A positive. So this is foreign to her, right? She's never seen this before, or her immune system has never seen this square before. So her immune system the immune system of the mother will react and will generate Rh antibodies. Right? During the birthing process, during labor. And now, after this first child, her blood is going to look slightly different, won't it? It'll have, of course, that same RBC will not change, right? The RBCs don't change, but her plasma will have changed. She will now have the anti-B antibody, but she will also now have constructed a new antibody with that square shape, and that's the Rh, right? And so now let's go for round 2, right? Let's say that these two individuals go ahead and have the second child together. Father, A positive, right? Second child. Let's just repeat it the same way. You can think of other examples, but I'm just going to go over this one example. Second child. Again, A positive. Let's go ahead and draw that RBC out of this child. It's got the A triangle antigen. It also has the square-shaped Rh antigen. And now we want to ask ourselves, has anything changed from round 1 to round 2? And of course, yes, there has been a change. And this is an IgG. And IgGs are small enough to cross the placenta. And so, right, if this right here is the placenta, we can now imagine a situation where the IgG does in fact cross over and enters the child's bloodstream. And now you can see how this would be a problem, because this is an attacker. It is an antibody, right? These are the proteins that attack bacteria that you manufacture against viruses. So these are attacking proteins made by immune systems. And of course, they're going to target this cell for destruction, and they will stick with their sticky ends onto that antigen. So you do have a match here if this happens. And if this is the case, RBCs will get destroyed inside this child while they're inside the womb, and what we will have is hemolytic disease of newborn, which is a type of anemia where there's not enough RBCs being made, or too many are being destroyed, I should say, and we end up having jaundice, which is that yellow complexion of a newborn when this antibody is present attacking its RBCs. So now that we know this, of course, medicine, technology is able to figure this out for us. So now what we do is we always take the mother's blood type, and if the mother is negative, right, if she has a negative type blood, then before the birthing takes place, before labor, we administer a RhoGAM, and RhoGAM is a type of medication that interferes with the production, right? It interferes with the production of this antibody. So this antibody will not be manufactured if we give RhoGAM to a type negative blood mother. And so therefore we then are not going to have to put the second child at risk of this type of hemolytic disease of newborn.